Hello, my name is Dmitry Kouznetsov. I am a PhD student at KU Leuven, Belgium, at the IMEC Research Center. I'll present to you my research on designing of optical lattices for lens-free fluorescence microscopy. The fluorescence microscope itself is arguably one of the most popular tools in the life sciences, and miniaturizing the rather bulky equipment to chip size might set it free from the specialized lab environment and make it available for all. We propose a concept based on integrated photonic circuits, co-integrated on a CMOS camera chip to avoid the complex free space light paths and the rather large multi-element lens-based light collection optics. Bringing the fluorescence microscope to the chip also carries with it all the applications that are possible using fluorescence microscopy. Since chips can be mass-produced, the on-chip microscope would enable high throughput for cell imaging, photochemistry, and next-gen DNA sequencing, among others. All modern imaging modalities rely on complex illumination patterns that allow for controlled optical sectioning, so as to eliminate the entire sample at once and perhaps get a blurred picture, but to build up the image from smaller chunks. I'm mostly interested in generating sparse arrays of excitation foci to enable a scanning-based approach. The objectives are therefore to develop a theoretical framework for such arrays, then validate its spatial properties, and finally translating these ideas to chip components that would be manufactured. I'll tell you a bit more why distinct illumination spots for scanning is crucial for a working device. The idea behind using photonic integrated circuits for illuminating the sample relies on exciting the fluorophores in the sample with the evanescent tail of the excitation light transported by the waveguide. This is really the principle of total internal reflection and would excite a thin layer of fluorophores close to the surface of the waveguide. The problem is now that when illuminating the sample uniformly, all fluorophores are excited at once, and the pixel array can no longer discern between adjacent features, resulting in a very blurred image. On the other hand, when using a sparse array of illumination spots to scan across the sample, the emission signal can be resolved in a time domain, and with that the resolution is now determined by the size of the spot, which is around 250 nanometers. Generating the spots themselves is rather difficult, since the geometry is confined to the plane of the chip. There were beginnings of a framework for generating sparse spot arrays in the theory of coherent optical lattices, but the results went virtually unnoticed in the scientific community, outside maybe of light sheet microscopy. So what we found is that generating these optical lattices in pretty much any laser beam configuration depends on prime factorization of integers. There is a bit of algebra involved, but geometrically, the picture can be understood as well. A regular grid, triangular one, is represented here by gray dots. One can then draw circles around the origin. And for some radii, the circle will intersect with a number of grid points. Some radii will have no points on them, some 6, some 12 or more. And then associating the angles of these concyclic points with beam angles and calculating the resulting interference pattern where all beams intersect gives you a coherent optical lattice. And is the distribution of the points on the circle that determines what kind of pattern you get, how sparse the spot array. I can show you more patterns here. Using the algebraic framework, it is possible to systematically find all triangular, square, or even quasi-periodic patterns. I was also able to experimentally validate some prime examples of patterns in a multi-beam interference setup using a binary amplitude mask where the aperture positions correspond to points on the same circle. You can also see by the yellow markings in the experimental data how different beam configurations affect the spacing of the intensity maxima. To conclude, a theoretical framework was developed to generate arbitrary, highly tunable illumination patterns in planar geometry, thus translatable to chip geometry, The patterns were also experimentally verified in a free space optical setup. What remains is to translate these beam configurations to photonic integrated circuit components, such that the desired patterns can be generated over large areas for a large field of view of the microscope. One of the challenges is that in the theoretical framework I assumed planar wavefronts, but real light beams are curved. In planar geometry, like on a chip, the light waves are actually cylindrical. So an expansion of the theoretical framework for different wavefront shapes would be a very important step as well. With that, I thank you for listening and have a nice day.